Welcome, everyone. Sorry for the delay. This is the second lecture of the 2022 Fishtails Lecture Series, presenting the science of Great Lakes fisheries. Tonight, we will have another engaging and informative presentation on Great Lakes Acoustic Telemetry Observation System, also known as GLATOS, Prying into the Secret Lives of Fishes by Dr. Chuck Krueger from Michigan State University. The Fishtails Lecture Series is an educational outreach program, the Crossroads at Big Creek Learning Center and Nature Preserve. And my name is Mark Holey, the Fishtails program organizer. I also want to recognize the Door County Library and thank them for their partnership to present these things live via Zoom and Facebook, and for those who can't be here in person. You know, I hope that you'll be able to join us on our two remaining lectures. The next one, Dr. Carrie Ann Heyer from the U.S. Fish and Wildlife Service, who leads their invasive carp environmental DNA surveillance program on Lake Michigan. And we'll discuss how to use that eDNA as a tool to detect the first arrival of invasive carp into the basin. And her presentation will occur on Thursday, February 24th. And to wrap up the 2022 lecture series on March 17th, Dr. Karen Murchie, the Director of Freshwater Research at the Shedd Aquarium, will present her research on white and long-nosed suckers in the Great Lakes. Dr. Murchie's sucker research spans across the Great Lakes Basin, but Door County tributaries are a focus of her area of research, and you will find her work on tributaries such as the Hines, Hibbards, Samuelson, and Big Creeks very interesting, and you will not want to miss it. A few housekeeping details for those who are joining on Zoom. Your microphone should be muted during the presentation. Please use the chat feature to ask questions that we will address at the end of the presentation. This lecture does include live transcription if you want to use it. And to access that, please click on the closed caption symbol in the bottom toolbar and then select Show Subtitle. And finally, this lecture will be recorded and will be available in the future at the Door County Library calendar site, the website. Now, why is that doing that?
We also have something.
What happened here? The best laid plans don't always work. And it's you.
It's all my
So maybe I'm sharing the wrong screen. Meeting controls. Okay, so turn that on. Boy, this is the strangest thing that's happened. Why is it doing that? Let me get rid of that. How did I get back there? Now what's showing up there now? The same thing, lead meeting. Yeah, I got too many things open. Okay. Yeah, I don't know what this is. Well, let me.
Oh, I get it. When you expand.
Oh, because I wanted to add a slide there.
Yeah,
I screwed it up when I did that. Okay, so now I'll go back here. How do I get back? Okay, then I gotta go back here and then I'll share a screen. And then I'll click the right one. And then I should be able to go like this. How's that look?
that looks good.
Okay. Okay, so then I should be able to go like this. Hey, we got okay. Ta-da! We got it figured out, all right? So our speaker tonight actually is one of my earliest Great Lakes colleagues. Both our careers in the Great Lakes fisheries started working for the Wisconsin Department of Natural Resources. And we overlapped for 2 or 3 years when he was the Great Lakes staff specialist in Madison. And I was a young biologist working on the commercial chub fishery in Milwaukee. After his stint with Wisconsin Department of Natural Resources, Dr. Krueger accepted a professorship at Cornell University in fisheries with a focus on applying the disciplines of population ecology and genetics to solve issues in fisheries management. During his tenure at Cornell, Dr. Krueger also served 10 years as a US commissioner for the Great Lakes Fishery Commission, and eventually left Cornell to become the science director of the Great Lakes Fishery Commission. Since 2014, Dr. Krueger has held the Thomas Waters Chair for Aquatic Ecology and Conservation at Michigan State University. In 2010, Dr. Krueger initiated the GLATOS program to bring collaborative acoustic technology tools and applications to the Great Lakes to unravel the mysteries of fish in the Great Lakes and to enhance our understanding of fish ecology and fisheries management. And not only is Chuck a prominent scientist in the Great Lakes, he also can catch fish. And this happened just this morning. Oh. Excuse me. And this was just this morning out with Nick Nault and also his dad, Gary, and brother Chum. And we had a great time this morning on the ice today. And with that, Chuck, take it away. You're probably gonna have to stand in front.
You gotta stand next to the microphone.
Mark,
Yeah.
your screen is not showing, it's showing PowerPoint, it's not showing your slides on Zoom.
It says I'm sharing my screen.
It's showing the PowerPoint view, it's not sharing the actual slides. If you look on Facebook, you'll see what I'm talking about.
So you mean you see the, does it show slide 3 of 88?
No, I'm not seeing that.
Got me. Hang on a minute. So we'll stop sharing here. What do you see now?
Now we just see all of the
You
gallery
see me?
view.
So you see me, right? So
Yes.
Screen share. That's where I want to be.
That's looking a little better.
Well, what's a little better?
It was showing side-by-side screens.
Got me.
Now it's showing
A full screen?
not full, but at least it's showing the slide that you want to show, number 3 out of 88.
Now, why would it do that? Hang on a minute. Anybody got a clue?
I think that would work better if you just want to proceed. I think that would work better if you just want to proceed.
Pull down display settings at the top and click presenter mode.
So what do you see? I'm going to stop sharing and try it again. So if we share screen,
Now it looks better, a little better, yes.
but it's got to be able to turn. Ah.
Yeah, that looks good.
I don't know which one we're on. My problem is I got multiple.
Number 3.
Number 3.
Try advancing that. I think that's going to be okay.
What?
Just try advancing that and see if it takes you to. Yeah, I think that's going to be okay.
Okay.
This is what they're seeing, maybe not a full screen, but they at least are seeing.
Okay.
It's small, but it's there. It's great.
Yeah. So let's let me. No, I'm just going to start all over. Yeah, you're worried about your dots, right? Right. But let me. So hang on a minute. We'll get there. So now I don't have anything open, so I'm just going to open one. Because we had multiple ones open, right? Right. So I'm going to do this. Although I got to share a screen. Well, yeah, that's okay. Right. So we'll do this. But then I can't get back to this share screen. I guess I can right here. I don't want that one. I want this one. Well, that's what they're going to get. I'm not going to do it again. So can you see something? I can't advance this stuff. There. Do you see the title slide, Laura?
Yeah.
Yes, we see the title slide in the next slide.
Well, okay. Here, I'll cover it up. So anyway, take it away. This one's full here, right?
Yeah.
Okay.
And we got all these wonderful people that came on a windy Monday night. Yeah, so I got to speak into here, right? Yeah. Are we using a mouse or am I doing it on the touchpad? We don't have to. I mean, I can do a touchpad, I guess.
Yeah, I just
okay. All right. Great Lakes Acoustic Telemetry Observation System. That's a lot of syllables, hard to say. So as Mark has already said, we often call it GLATOS. So, but really what this is all about is indeed prying into the secret lives of fish. Let's see here. Whoops. All right, so what I'm going to talk about today, I'm going to back out of acoustic telemetry and first just talk about tagging and marking fish, okay, and look at a couple example tags. Then look at the question, why would we want to tag fish? What types of information can we get from tagging or marking fish? And then what is acoustic telemetry? So kind of narrow down off of just generalized tags and marks and talk about a very specific type of tag, one that emits sound. And then talk about some real data, some data that we've actually collected. Where did the fish go? And we'll do a comparison of Lake Huron, and Green Bay. And we're just going to look at walleye. We got lots of data on other species, but I thought maybe walleye would be of interest here. I did get a specific question in preparation because we're going to change the receiver pattern in Green Bay from going lines across the lake, different spots, to a gridded system. And I'll try to explain why. And then last, well, so we collect all this data, costs a lot of money. Is it ever used in decision making? So have the data been used? From my perspective, at least for the audience that's here, if you've got a question along the way, it might be most effective to ask that question right away, because you'll probably forget at the end. So that'd be my preference. So we get into the tags or something like that, you can feel free to ask questions. Just raise a fin and let me know if you want to speak. I thought you were going to sit back. How about if I did this to Chuck? You can take a picture of me at some point. All right. Thank you. Yeah. All right. So onward. So first, what is a tag and what are some example tags? You can think about tags as being advanced pretty quick, as doing a mark or an attachment, something placing on the fish or within the fish that doesn't occur on the fish, something that's recognizable. So it's used to identify one of two things, either groups or individuals. So a really common mark that fishery biologists use is clipping a fin. So if you clip a pectoral fin, that's typically a permanent mark. And of course, it's just one single mark of no distinction. So that's only going to do groups of fish. So you might do fin clipping of a raceway where all the fish were raised in a common raceway in the hatchery right before stocking. That would be an example use. External tags are something that we might place on the fish. And so a couple examples are jaw tags that go in the jaw, obviously, and spaghetti tags. And we'll take a look at that. And then last is internal tags. These are put in typically surgically, and they emit some type of frequency, so radio frequencies or acoustic or sound. And so the information is provided when the fish is either recaptured or observed in some way and the tag detected. And then you know where you tagged the fish and where you recaptured or observed it. And then you know the time span in between. Okay. So some example tags. This is an external jaw tag from Lake Erie on a walleye, and Ohio DNR has used these for decades. And in this case, if you can see that, it's you get $100 if you return that to the Ohio DNR. So it's kind of like a prize winning, and it's got numbers on it. So that would be an individual mark. So you could go back to that serial number, see what the weight and the length of that fish was, when it was marked, how much time has passed, and how that weight and length may have changed. An external tag, we call those Floyd tags, dart tags, spaghetti tags, various types. A common type is one that's used in the garment industry for putting price tags on. You've probably seen that on a coat, and they got a T tag. So when you put these in, if you can see that green tag on the rainbow trout there, it's right below the dorsal fin, all right? So if you flayed a fish, you probably know there's bones that come down below that dorsal fin, and you stick that tag in, turn it that way, and then that teeth pulls up against those bones. And those tags can stay in a surprising amount of time. And with that, I have a couple tags I thought I think I'll wait on that. Whoa, that's amazing. Okay. Why does it do that? I don't know. It just went forward about 1 million. Okay. Okay, so why would we tag fish? I mentioned that we can get movement out of that between a release to a recapture location. Obviously, you can get also behavior, because if you look at where you caught the fish or observed that fish selected to be there. So that tells you something about habitat selection. And then in terms of population dynamics, you can estimate with some statistics, both survival rates and abundance. And this is through like mark and recapture. You look at the proportions of marked fish in your recapture sample. So all this information is important for fishery management decision-making. It can advise the setting of regulations, where to protect habitat, and some stocking decisions. Okay, so we'll narrow in now on acoustic tags and what is acoustic telemetry. In a nutshell, acoustic telemetry involves then the placing of an internal tag surgically. That tag emits an acoustic signal. What's unique about that is every acoustic signal is indeed unique. So in other words, it's an individual mark. So, you know, Tom, Dick, Harry, Sue, you can tell that those fish by individuals. That tag swims around, or the fish swims around with the tag. Tag doesn't swim, but, and it emits that unique signal. Typically, we set them up to transmit every 2 minutes. And it'll do that. Some of our tags will keep going for 10 years. So 2 minutes every day, 24 hours a day, 7 days a week. Fish swims by a receiver that's most often anchored in some manner on the bottom. And if that transmission is within range of the receiver, that unique transmission is decoded, and there's a date and time stamp that's put into a data file. And you know where that receiver is placed, so you know that location. And then you have to come and retrieve the receiver and download the data, okay? So it's not a real-time data situation. You come back a year later and find out what fish swam by your receiver. Don't do that. Okay, tags. They typically transmit at 69 kilohertz. You don't want to put a tag in so heavy that it sinks the fish to the bottom. So our general rule of thumb is 2% of the body weight. That usually means that the smallest fish that we can tag is around about 1/2 pound. There are a variety of tag sizes, as you can see in this picture and then in the next slide. I think what's cool about them is that they are programmable. So you don't have to do 2-minute transmissions. You can do 5-minute and extend the battery life on them and extend the life of your tag. Or if you're not interested in, say, winter data, you could have the tag turn off from December 1st and turn back on April 1st, saving a lot of battery life and again, extending your tag life. So the life varies on the real little tags that have real little batteries. It's all about the battery. It's only a few months. And you can tag small fish, but you don't get anything more than that. On the big tags, in this particular vendor, the V16s that we put into lake sturgeon, we get 10 years out of the tag. So it's a pretty cool system. Range on the transmissions for detection range from about 1,500 feet, 1,600 feet, up to 1.3 miles. Now we have detected tags more than 7 miles away in some of our testing under ideal acoustic situations. Okay, here's just an example of a V16. I'm going to send these around. So there's a couple tags now on these tags that I'm show and tell here. They have magnets on them. So don't take the magnets on, that activates the tag. It's got a magnetic switch. And then there's a type of a spaghetti tag that's coming around. So I mentioned this is done surgically. Most commonly it's from the abdomen. And so we have these cradles made up, water going into their mouths. They're under some sort of anesthesia. And here's a B16 being put into the abdomen of a lake sturgeon over on the St. Clair River. Throw some sutures in on the incision. The yellow stuff is betadine. That's just a disinfectant. So on these sturgeon, we typically put in three sutures. Okay, so after that's done, then we let the fish recover, usually in a tank, but it's try to minimize the amount of time. As soon as they look like they're good to go, over the side, back in the water. On the sturgeon, we have virtually no evidence of any post-tack mortality. Receivers, this is what a receiver looks like. Typically, they're stationary, they're not being moved through the water, they're fixed to the bottom, anchored in some way. As I mentioned, the data are downloaded upon retrieval, so it's not real time. Now there are, yes, we have them on the side here, in my box here. You can put a receiver on a boat, move the receiver through the water, so that'd be like active tracking. There's some of that is done. The best example I know of is over the Mississippi River, where they put these receivers on barges running up and down the river, and they were detecting carp. We have a real-time example. In the Sandusky River, we had a receiver that was connected to a cell phone network, and you could actually look it up and see what had been detected most recently. And that was part of a grass carp program. So receivers coming around. It's a little beat up, but you get the idea. So receivers decode the transmissions and they add a date and time stamp. A lot of times, all over the country or in saltwater, the receivers are placed in lines, and that just gives you a sort of a place of detection. So it's really kind of like a one-dimensional detection. But you can put receivers out in grids, and that will either give you that one-dimensional detection, I'll show you a little bit of that, or you can get 2D detection. So in this example, I have a lake trout that's pretty close to the receiver at the bottom. And if you have those receivers, their clocks perfectly synchronized, so they're exactly the same time, then you can look at the time of detection difference between that receiver and the receivers that are further apart. You do a bunch of fancy math, and you can come up then where it was in that triangle. And so then you get actually a two-dimensional sort of position on the fish. Our receivers anchored. Now this is really high tech. The anchor here being used is concrete poured into the bottom of a garbage can. It worked really well. And we just got a simple slide that we just push the anchor off. That's sort of what we call flying saucer looking thing above him is a fancy GPS unit, super high precise, you know, down to the nearest meter or something. And so wherever that anchor goes in, that's recorded. So we know exactly where that receiver, and that can be pretty handy when you start losing receivers, you need to go find them. Here goes the anchor. There's a buoy that was above the receiver. That's to hold the receiver upright in the water. And then attached beyond that is then a flag in this case. The flag buoy is used then to recover the receiver. You can just pull it from the surface. This works really well where you don't have a lot of the public going around there. If you put a buoy down like that and people are out there, somebody will come over and pull it out. So this only worked. This worked real well over in Alpena in Lake Huron, but it was October during lake trout spawning season. Pretty much everybody's gone. And there's the flag buoy in the water. And then this is a picture just of being in the water. You can see how the receiver is held upright. That assists in the detection and recording. As I mentioned, of course, you can't always have flag buoys. So a lot of receivers, and some in Green Bay and quite a number in Lake Erie, have acoustic releases. So that's in this case represented by this yellow thing that's next to the receiver. There's two buoys that are affixed to it, and then it's just anchored to the bottom. And so in this case, well, you have a detection range. Fish swims, supposed to swim by, there it goes. Swims by, transmits, and then we send a specific acoustic signal down, and that releases that acoustic release, and the receiver goes to the surface. And this works really, really well for recovery because you can be out, I don't know, 1,000 feet or a little less, but you can be motoring up to the area before you even get there. It's setting that signal so that by the time you get there, the receiver's on the surface. So you can tend a lot of receivers in a hurry. Okay, so what causes problems with tag detections? Well, essentially anything that makes noise in the water. So if you got a whole bunch of fish right next to one receiver, and they're all transmitting at various intervals, so you can get then overlapping tag transmissions. So that'll confuse things. Sound that comes from wind and waves. Obviously rapids make sound, but they also have air entrainments. They get little bubbles in the water, and that affects then how the sound gets transmitted through the water. Anything that changes in density, so like thermoclines or temperature stratification, things that can absorb sound, like algal blooms or vegetation. You might think in the winter, if you had ice cover, that'd be great, really quiet. Problem with ice cover is hard. And so the transmissions echo off the surface of the ice, and that can cause troubles. Just like substrate type, hard versus soft, you'd think maybe hard would be good, but actually soft is better because hard substrates cause echoes as well. Ship engine noise, probably obvious, but there's a lot of noise in the water when a ship goes by. And so like in the Detroit River, St. Clair River, where we have a lot of shipping, we get periods of time when it's just blanked out. And in saltwater, there's a lot of animals. I don't know if drum really make a problem, but certainly shrimp do. They make noises and can block signals. Now if you're doing a project like this, there's some issues with this because other people are going to have receivers in the water. And you putting tags in fish, and your fish don't know that they're supposed to stay over here, but they swim over to this other guy's receivers, another project's receivers, and they record your fish. Well, you'd like to have that data. So data sharing is a big deal, because then you can access all the receivers that are in the water, not just your own projects. Now, if you know scientists, they're not always the happiest to share data. So there was a bit of a cultural shift that we had to do in the last decade to get scientists to be willing to share and partner across. And that's one. And then if you do that, you also have to know about their receivers, where their receivers are, and when they're tended, and when they're actually operational. Fisheries are underway, and we'd like to get tags back from the fisheries. So all of our tags have some sort of label on them about contacting and sending them in. They get some sort of reward. But those are actually valuable data, because if you get a high enough return on your tags, that's why we offer a lot of money. You can actually then calculate things like fishing mortality, not just total mortality. Okay. And obviously, to pull any of this off then requires communications among the investigators and between investigators and the public. So that's why we created the Great Lakes Acoustic Telemetry Observation System, or GLATOS, which might be the Greek goddess of acoustic telemetry. And the purpose of GLATOS is certainly conservation management of fishes, but it goes after those main sort of topics like fish behavior, movement, habitat use or habitat selection, and then population dynamics, where you can do survival and abundance. Some questions we've been after in behavior is what's the contribution of Lake Erie walleye up into Lake Huron into Saginaw Bay? There's been a lot of thought that with such a big population in Lake Erie, these were bleeding off up the river and going into Saginaw Bay and actually being the main source of the fishery. Another one was what's the critical overwintering habitats of lake sturgeon in that Huron-Erie corridor, Lake St. Clair River, Lake St. Clair, Detroit River. Indeed, in that case, we found that though there's 2 or more populations of lake sturgeon in that system, in the winter, a whole bunch of them all go to Lake St. Clair. So if you had an engineering project for dredging for the shipping industry, you could have a real problem in Lake St. Clair if you started messing around with that a lot. Could be a problem for the sturgeon. And last, annual survival rates. I mentioned you can calculate those. Those we've actually done for lake trout up in northern Lake Huron, associated with the Drummond Island Refuge. So that's GLATOS. Where are we now? We have projects in all 5 Great Lakes and in the interconnecting waterways like the St. Mary's River, and here on Erie corridor, St. Lawrence River. We have 43 projects, 49 species have been tagged. There's been almost 17,000 receiver deployments, but we have in the water now about 2,000 receivers across the Great Lakes that are active. More than 15,000 fish have been tagged with acoustic tags. And in our database, we have over 537 million tag detections. So an investigator can go with their tag IDs and search that database and see where their tags have been detected. Oops. It's a little touchy. Okay, where did the fish go? So what are some actual data? So we're going to use, let's do this. There we go. We're going to do a walleye example. Saginaw Bay, Lake Huron, so it's in the southwest corner. If you're not familiar, it's shallow bay. Pretty good walleye fishing. It's a shallow bay. Walleye is a really big deal with it. Well, okay, here they go. Well, you can watch this. That's Saginaw Bay up there. Where that blue triangle, where that red dot is where we tag the fish. About over 200 walleyes. And if you know, this is some fish stay in the bay, other fish go to the right, other fish go to the left up to the bridge. And this goes through several years of data. What's really cool, I think, about this is we know these by individuals. If you were a left-hand turning walleye last year, you're a left-hand turning walleye the next year. If you were a resident fish in Saginaw Bay, you stay in Saginaw Bay every year. So it's by individual. It's a very consistent pattern. It's repeatable over time, regardless of climate and weather you had in a particular year. What's cool, if you were a fisherman, I think there's a couple in this room. Half the walleye leave Saginaw Bay in the summer. Okay. The other half come back around Halloween. So if you weren't a deer hunter or a duck hunter, you could go fish Saginaw Bay around Halloween, first week of November, and there'd be twice as many walleyes in the bay at that time than there was in the summertime. Not that I would ever think of such a thing. All right. Yes, sir.
So the left turning and right turning walleye.
Yeah.
Have you detected any genetic differences? I mean, is that one of the studies?
Yeah, what the question is are there genetic differences between right and left-hand turning and resident fish?
Right.
We don't know that, and using neutral genetic markers, it would be very difficult to see that because these were all tagged below the dam on the Tittabawassee River, presumably representing one population. So we don't know. But my theory is a fish goes out there and for whatever reason, one day decides to turn left, goes up to Alpena and Thunder Bay and finds a lot of food, thinks this is a good deal. And then goes back to spawn and comes out and goes, well, I'll do that again. So I almost think it's environmental, experiential than genetic, because they're a common gene pool that's just sharing right below the dam there. But I may be wrong. Okay. So Green Bay, I'm just going to show you a little Green Bay data. So this is very early, forced people to put this together. Many thanks to Todd Hayden particularly. So there was, and Eiserman too. 79 walleye tagged up in Little Bay Denok, 93 walleye in Southern Green Bay. So there's more walleye tagged down in Southern Green Bay than Little Bay Denok. Some of the questions we're after is, how much does Little Bay Denok or the other areas there contribute down into Southern Green Bay? And so then the gold dots are receivers. So those are places with some ranges around them to tell where the fish have been detected. So what's going to happen is this next one is going to have the bay change sort of like colors, and it's going to represent seasons. So if you think of the pattern back in Saginaw Bay, they all came back in the fall and they all blow out of there as soon as they spawn and go out into the bay. So I'm interested to see if these walleye do any of the same things. Okay. Stand by. Did not. There, that's. Chuck was going to do a dance.
Yes, I was in.
There it goes. Okay. Winter, the bay is dark. Spring, it's light. Now we're tagging fish, and you see them pour out of Little Bay Denok. Some going out into Lake Michigan. A few going down in Southern Lake Michigan. But remember, we had more fish tagged in Southern Lake Green Bay than we had up in Little Bay Denok. Not many detections from Southern Green Bay. And of course, as you get into the autumn, look, they all go back to Little Bay Denok. So a very similar pattern to what we saw in Saginaw Bay. There they are, springtime, spawn out of there and disperse. But again, are just not seeing much down in Southern Green Bay. So we don't know if that's caused by post-tagging mortality, or we just didn't have receivers in the right place. You want to see that again? Can you see it?
Yeah.
I mean, look at the fish that are in Fox River. And they sort of blow out of there, and then that's the last we'll see of them.
None go through the canal. Oh, one.
I think it was one or something. Yeah, we have receivers in it now. Because I thought, oh, maybe they go through there. We see all the receivers. Yeah. This project is serving not just walleye, but there's whitefish and sturgeon. There's been muskie stack. No, these were tagged at some bay sites. Yeah, but I mean, it's all in southern. All right, so, well, we got that to work. So the question then, we're going to change from lines and go to a grid pattern, and maybe obvious why. This is the former system that we had, Chambers Island, and off Canal, Sturgeon Bay, Ship Canal, and off the gap off the end of Door County. And then lines, little big bay to knock and little bay to knock. So we're going to take those receivers and spread them out. Our main questions were the differences in use between North and South Green Bay. How many fish leave Green Bay? What are the contributions to low bay to knock, big bay to knock to Southern Green Bay? But we have some data now that really tell us about those questions, but moving to this sort of pattern will still allow us to address those questions over the years. And it should solve this problem in Southern Green Bay as to what happened to those fish, because they're not going to be able to get through that. They'd be spaced about 5 miles apart, something like that. And then the other thing is that we have had other projects in northern Lake Michigan go in the water with receivers. So this is a picture. These receivers are in the white dots are Lake Trout project, and the yellow and the orange dots are Lake Cisco or herring, depending on what you call them, projects on the near shore. So if we have some of our walleyes going out into the main lake and they go over to Beaver Island or those areas, we should be able to detect them. And with our GLATOS system in place, the investigators will be able to know that their fish were actually detected over on that side of the lake. Okay. So you got all this data, have they been used? So I've got 4 examples. First one is from Lake Erie. I think really cool because I've always been into lake trout. Lake Erie had a commercial fishery in the 1800s in the eastern end of the lake where the deep water, cold water is. And there's been some efforts for stocking lake trout, and a lot of people felt it wasn't possible. There are adult hatchery fish that have grown up and are now mature, swimming around the lake. What are they doing? Are they spawning, or that sort of thing? So what's the question? So they put in essentially like a 2D type grid, and they could then actually triangulate where these fish went during spawning season. And based on that information, they were able to put down target fry traps right in spots where they thought they probably had spawned. Telemetry won't tell you if they spawned. They'll just tell you where they are. So these are pyramidal traps, if you can see that. And they have a steel frame on the bottom, mesh cone, mesh sort of triangular thing up to a collection bucket at the top. And then you just throw them overboard. There's a rope and buoys. The fry are in the bottom in the rocks, and at night they'll come up and get funneled into that trap, into that collection bucket at the top. So here, this is just their fry trapping. You can see they're pretty densely clipped down in one spot. And New York State Department of Environmental Conservation announced in August of last year, first documented lake trout reproduction. Indeed, they caught lake trout fry in Lake Erie. So that was pretty cool. And it did say that indeed lake trout can do the deed and actually make babies. Now, whether or not those fry survive, we'll have to see. But it could be the first start of natural reproduction in Lake Erie. Lake Erie grass carp removal is kind of a unique sort of use of acoustic telemetry where you tag a fish that serves as a Judas fish, if you will, that then tells the location of where other grass carp are. And if you've got that information, you can send out the electrofishing boats. Ontario is particularly keen about removing grass carp from Lake Erie. And indeed, they catch some grass carp by using information like that. In terms of walleyes, Ohio DNR is keen to have a fishery in April during the spawning season. People fish them on the spawning reefs over in the Western Basin. There was a big concern about, fishing's pretty good and whether or not that would actually affect recruitment or spawning by over harvest. And lake trout or walleyes were tagged and they were watched as to how much time they actually spent on a reef. And what they found was that males spend most of the time on the reef. They're the ones that are vulnerable to angling. And the females, which primarily drive natural reproduction, they just come up the reef, do their thing, and they're out of it. So gave some comfort to allowing sport harvest during the spring spawning period. And then in Saginaw Bay, which I've already mentioned, because half the population moves out of the bay, they liberalize bag limits on walleye out in Lake Huron proper. But what about in Green Bay? Now it's pretty early for application. I really try to caution people of jumping the gun on the data. I always feel like you should see 3 years of consistent patterns. So you get some changes in ice cover and weather and all that sort of business. If you see the patterns, it's happening every year consistently, then it's time that you could actually apply it. There was some use of it already, whereas the southern Green Bay lake whitefish appeared clearly separated from the main basin population. And that supported then some of the proposed changes to whitefish harvest management in Wisconsin waters of Lake Michigan. And based on some muskie tracking, where the adult muskies went, identified some other habitat areas that looked like they'd be better for juvenile survival. And so the state of Wisconsin actually moved some of their stocking sites for muskies based on some telemetry data. So that's kind of the first uses over here. So in summary, okay, tags are used to identify groups or individuals, and tags can be either anything from as simple as a clip fin to electronic transmitting tags, radio or acoustic or sound. Acoustic tags, typically we set them up to transmit every 2 minutes. And they have a unique code for each fish. Receivers listen for the tags 24 hours a day, 7 days a week. We have to usually go out about after 1 year's time and then recover that receiver, download the data, and put new batteries into the receivers. Studies have been or are being collected, are being conducted on every lake for a wide variety of species. And managers have already been using these data in decision-making, including for regulations and stocking. So I'd be happy to answer any questions if you have some. A little jerky on the slide, sorry about that.
Questions?
Question over here, yes, sir.
One thing I know you didn't touch on.
Price?
Can you tell the group about that?
Sure. Tags cost, depending on the size and how you set them up. You can actually have tags that, I was going to send some new tag stuff out. You can actually have them set up to tell you depth based on water pressure. So all those sorts of things vary in the cost, but tags are, say, $200 to $500 in that range. Receivers vary, with basic sort of receivers at $2,500 to $3,000. But they have integrated acoustic releases on these receivers. Now I showed you one where there was 2 separate units, but they have one that's now like it's all put together. Those are almost double the cost. Yeah. So it always sounds like a big upfront cost, but if you look at the backside of the information that you get, compared to if you were doing just traditional marking with fly tags or something, it take you 20 years or more to get to where we are in 3 years with this. So there's a time element. So I always feel it's hard to do a cost benefit analysis comparison with other procedures. Partly because the quality of the data is so high and it's so quick and reliable. And you get multiple observations on the same fish, which you wouldn't get on a lot of your marking techniques. You only get one recapture typically. Good question. Yes.
Would you say that the main goal of this is to benefit fishing, or is it conservation?
Yes. No. Well, it's not to benefit fishing. I mean, the point of it is not to necessarily, maybe as a byproduct, will tell anglers where they might fish. But the benefit is for conservation and management, so that you can have good regulations or protect habitat where you need to. Yeah. Yes.
So just to extend on that, you saw that receiver array up in northern Lake Michigan around Beaver Island. So that's actually evaluating the efficacy of the refuge, the fish refuge that's up there, that's used to protect lake trout that are spawning there from fishing. And we're actually testing that hypothesis. It's been there for a long time. Some people wanted to go away.
It's very similar to the project we had on Drummond Island, which is also a refuge, a seasonal refuge there. And yes.
Well, the Mink River's an estuary too, isn't it? In the spring, you cannot go in there and fish for a certain length of time.
Where's that now?
The Mink River.
What's that? Oh, okay. Yeah, I'm not familiar with that.
Okay.
So Mink River?
Yeah. Never
Okay.
heard of it?
No. I'm honest. Right?
But there aren't any studies being evaluated on the Mink River right now.
Not that I'm aware of.
Could be.
Are there any studies evaluated on the Mink River? Was the
Well,
question.
2 years ago, I read they were doing a northern study in the Mink River to see.
Well, when I say study, there aren't any acoustic studies. It's a great mistake. There's people who have worked there occasionally, but not with the tank just yet.
So just repeating there are studies on northern pike being done there, but not with acoustic tags. Yes, sir.
The retrieval of the receivers. I noticed that is it cell phone operated? The ones that release.
Oh yeah, acoustic. Yeah, acoustic releases. Yes.
The ones that you pull up from the floats.
Yeah.
You disconnect them from the anchors before you pull them up. You pull up the whole thing.
Typically, we have like a davit off the boat, and that you pull up the whole thing. Yes. Yeah. So the question was, do we disconnect the anchor from the receiver when we pull them up with floats? And no, we pull up the whole works with the floats. Been a bit of an issue over on Isle Royal where we're doing a lake trout project. There we're using natural rock because we're using acoustic releases there and we're leaving the rocks behind. So we actually have rocks, natural rocks that are the anchors so that we're not leaving concrete in the water. Yeah.
They manufacture batteries for the research on mammals.
Well, there's lots of radio telemetry on mammals, and there they have batteries, of course, for those too. Those will actually uplink the satellites, and they will actually contract the mammals. I'm thinking like caribou in Northwest Territories, they have radio collars on them. You can get on a website and find out where the caribou are.
You know, wolves and
Wolves
and other species.
Yeah, wolves, bears. They even have little tiny radio transmitters they put on woodcock, little saddle things that go around the wing.
What can you say about
No. Can I say anything more about data management? You have to have a data manager to handle this because it's huge. I mean, if you saw how many pieces we have. This is not something you do on an Excel spreadsheet or anything. So we have a full-time. I actually made sure that we hired a data manager to help set up, and then we have another person that really helps with just dealing with the website. And then we got an external website contractor too. So I knew at the front end that this was going to be a huge issue. I saw it with coded wire tagging on the West Coast where they didn't plan around data management. Yep. No, you have to be a member of GLATOS. But it would, I don't know what good it would be because you can look at a tag code, but you wouldn't know where it was from, where it was tagged. Oh, yeah, we have on the GLATOS website, we have some animations there you can look at. And Chris Vandergood, if you're listening, you better make sure it's there. Yes, sir. On salmon. So the question is there any research planned on salmon? Not that I'm aware of. It's possible that it's out there. There's acoustic telemetry done on Atlantic salmon in Lake Ontario, or there has been. But not that I'm aware of on Chinooks or coals or things like that. But probably there has been. Like I said, we have, 49 species. I mean, you can't, carp, freshwater drum. I mean, there's a whole bunch of things. Channel catfish, all sorts of things have been tagged.
The maps that you showed with the movement of the fish, are they available on your website?
Not the Green Bay one. The Saginaw Bay one might be. Well, you go over to Saginaw Bay. So the question was, are the animations on the website? And there are animations on the website.
I saw several of the Green Bay fish end up in sturgeon.
Yeah.
And you only tagged 93 fish, so there's probably 10 times even more. You could use that same number to figure out that maybe just one fish. Your dot showed one fish in sturgeon. That could have been quite a few more.
Sure.
The total population of fish.
So, yeah.
So if a guy can look at that map and then really put the time on it, target where would probably be the best place to fish walleye.
That's possible that you could do that. So the question was whether the animations are available and whether you could target where to fish. And to some extent, you could do that.
But you remember that it was only for that particular year on that particular date, and they weren't the same both each year that it showed.
Yeah, well,
so
the thing is, on Green Bay, we haven't. There is no data analysis at this point. This is the first any sort of summary of data. If you think about that, this is summarizing millions of data bits in space and time, and you're seeing it in like 1 minute. So, that's as far as we've gone with the data at this point.
One of the things we noticed right away is the green dots on Little Bay did not go much past
Chambers Island.
And the blue dots didn't go much north of it.
Right. It doesn't look like there's a lot of trading back and forth. And that was one of the questions. How much is Little Bay Dinock and Big Bay Dinock contributing to Southern Green Bay? Right now, it doesn't look like it's that serious. Yeah.
The people fishing north of Chambers Island are really more so chance to catch Bay Dinock fish than they are.
Yeah. Agreed. Yeah.
I got one more though. I use the paper punch method.
On the caudal fin, on the tail fin?
I do it on the upper lobe of the tail.
Yeah.
And I find that very useful for my, that I know if I'm catching the same fish every day.
Sure.
And
so the answer is?
Does not happen.
Yeah.
Even though I go back to the same spot with the same baits and everything, I do not catch many of the same fish. And I can still age those fish when I see one that the hole is completely filled in.
Yeah, you could see.
It's got a black meat in it.
Yeah.
I know I caught that fish 3 years prior to that.
So the comment is that you can mark fish instead of clipping a fin, you can do a paper punch and a caudal fin. They'll actually grow back together, but you could also see, not that I would ever do something like that, but in steelhead, we have done that in the morning on steelhead. We caught the same steelhead in the afternoon in a river.
I don't find that on smallmouth to be the case.
Yeah. All right, good. Well, thanks.
Wait.
No, what?
Let me double check.
Oh, there might be other questions.
Well, I looked through the chat there at the beginning. How long is the receiver at the same location?
Typically 1 year.
Typically 1 year.
Yeah, I mean, we've had specialized projects at Drummond Island where we've put them in August and we pulled them in mid-November. It was only a part-year project.
Okay, let's give him another round of applause. Good job. Thank you very much, Chuck. And just a reminder, if you want to hear about eDNA surveillance in Lake Michigan, come back on February 24th. That's a Thursday. And thank you very much. Good night.